Oh, bloody hell, that's loud. Good morning, everybody. And you can see there's Snowball. Keeping an eye out for, I don't know, postman probably. Doop-a-doop-doo. Buenos dias to you too. All right, let's see where we are at. Trying to remember where we left off from. Funny, I was actually just reading up on different models of homologous recombination, which gets pretty wild and wacky. Let's get that down there. There we go. Hey, you know something really cool? We'll get to this in a little bit. But actually, I have to take you back here. I don't know, for the ladies among you, this is probably more pressing than for the guys. But does anybody recognize those two genes or proteins in this case? These two here, BRCA1 and BRCA2. They are indeed, Desiree. They are, well, they're genes involved in DNA repair. And so if you have mutations in them, depending on whether you have mutations in both genes, it's a very strong predictor of breast cancer. Why breast cancer? Why breast mammary tissue and not other types of tissue or organs? I don't know, but particularly in those. And actually, I think it was Angelina Jolie, the actress and UN ambassador and stuff. She had a double mastectomy because she had a familial history of breast cancer, and she was homozygous recessive for both of those genes. And so her odds of getting breast cancer was something in the 90 to 95% range. So she preemptively had them removed, which is kind of crazy when you think about it. Yeah. So, but that just goes to show. But anyway, I thought that was interesting when I was kind of poking around in here, having a look at this. I was like, ah, there you go. Anyway, let's get back to business. So I like talking about stuff like that because it really shows you that we're not studying things which are just isolated facts. They are very much part of the bigger picture, even if it's sometimes hard to keep that in mind. So we finished up with the initiation of, well, we haven't quite finished the initiation of DNA synthesis, right? So this is again in prokaryotes. There are some added wrinkles in eukaryotes, which we'll get to in a little bit. But this is in prokaryotes. So we have the initiator proteins binding, open up the DNA. We have single-stranded binding proteins, hold the DNA open, right? We have helicase that further unwinds the DNA, and then DNA gyrase or topoisomerase, which relieves supercoiling ahead of the replication fork, ahead of that unwinding. And so essentially here in this image, if you kind of like have a closer look, You can see that this is the original template DNA, and the new DNA is being synthesized behind the replication fork. That's what this red ribbon is in this diagram, right? So that essentially shows you, without all the other stuff in there, what is basically going on. Okay, so anybody got any questions so far about any of that jazz before we move on? All relatively straightforward so far. Okay, so one of the biggest problems in, not the only one, because there are a bunch, one of the biggest problems in DNA synthesis and either prokaryotes or eukaryotes is the fact that you need a 3' OH, right? You always need a 3' OH to add a new nucleotide to, right? And so if you look at, once you've, let's go back up here. Okay, so in here, all we have is single-stranded DNA. There is no little bit of DNA here to provide that 3' OH for DNA polymerase to add a new nucleotide to begin the process, right? So if DNA polymerase can't do that, can you think of what enzyme can synthesize a nucleic acid using DNA as a template from scratch? Just have a think. This doesn't necessarily have to be in DNA replication only. mRNA, so what synthesizes mRNA? If mRNA is an RNA polymer? No. Well, yes, polypeptide chain does, but I want the name of it. If mRNA is an RNA polymer, the enzyme that makes it will be, well, yes, in this case, it's a polymerase, right? So RNA polymerase. Right, and primase is an RNA polymerase, right? So the deal here is, rather than making DNA to begin with, we make a little bit, 17 to 20 nucleotides long, of RNA. This is called a primer. It's very much the same as, you know, the primer in a bullet that initiates the larger explosion, you know, reading primers for little kids that get them going on bigger books, stuff like that, right? It's something that kicks off the process, right? And that's what's shown here. So the sole purpose of this RNA primer is to provide a 3' OH. So the sole purpose of the primase dang, I can never see my full chat box. Get over here. There you go. And this has to happen every single time synthesis of the daughter strand there is begun. So for the leading strand per replication fork, right, not bubble, but fork, how many times do you think this primase would need to make a primer for the leading strand? For one replication fork? Once, right? So there's only one leading strand per replication fork. How many times do you think this will have to be done for the lagging strand? And this is a little bit of a trick question. It's impossible to say how many times. It depends how long that replication fork goes on. Could be hundreds, if not thousands of times. depending on how far that replication fork travels and how big those Okazaki fragments are. Right? Because for every single Okazaki fragment, you need a primer. And so essentially what you end up with is this short, high teens kind of nucleotide long RNA bit. So you actually have an RNA DNA hybrid. So keep that in mind. We're going to have to fix that later. And then as soon as that primase is finished and presumably releases the single-stranded DNA, RNA polymerase can come in and it starts synthesizing the daughter strand, the new strand. And so the primase and helicase actually forms a complex at the replication fork, right? So you're going to have two of these per replication bubble, essentially. So as the helicase is unwinding, primase for the lagging strand at least, will be making a new primer, which you can see in green, which the DNA polymerase uses. That strand is then released, pulled back in, a new primer is made, DNA polymerase synthesizes another short stretch, DNA is released, pulled back in, and so on and so forth. Every single time an Okazaki fragment is made, the primer has to be made to start it. And this is going on in both directions, right? Unless we're talking about unidirectional replication in bacteria. So put a worm in your ear. Are we an organism that has RNA in our DNA? Does our genetic material in terms of what we pass on to children consist of RNA and DNA? It doesn't, right? So although that primase or the activity of that primase has solved one problem, no, just not at all, right? Even prokaryotes are DNA only, right? RNA is an intermediate for either translation or whatever else RNA needs to do. What's passed on to the next generation, whether bacteria or humans, is DNA, right? So this solves one problem, it creates another one, right? So when we talk about termination of transcription, like the very end, this is one of those problems that we're going to have to solve. So we'll get to that in a bit. And so it's actually kind of neat. One thing I've been thinking about a bunch, and you kind of come across looking at different mechanisms, is that all of this stuff is very ancestral, right? If you think about it in evolutionary terms, most of this stuff, there are like wrinkles in eukaryotes, obviously like extra differences, but most of this stuff is common to all DNA-based life, right? So even like the mechanisms of DNA repair and stuff that we'll be looking at in a little bit are deeply ancestral, right? So these things emerged really, really early on in the evolution of life on this planet. So it's just a really kind of cool thing to think about from that perspective too. Okay, so that's the whole process of initiation, open up the DNA, hold it open, unwind it, synthesize a primer. Those are the 4 kind of main steps. Actually, I probably better write that down. What's next? Oh yeah, unwind it. And then you got to synthesize. Ooh, that's not how you spell that. Prox is short for prokaryotes because I'm lazy, don't want to type that out. So those are all the main steps in initiation of DNA replication. The rest of it, actually, the synthesis of DNA is fairly uneventful, I would say. It's actually not very interesting, in my opinion. Some people do find it interesting. So all we do is you have DNA polymerase, which is not shown here. It's going to be a large complex of proteins to form that enzyme. All it does is match the incoming nucleotide to the base in its active site, like it has a pocket within the enzyme where nucleotides are allowed to enter. That match is carried out by hydrogen bonding. And so you have to have the correct number of bonds and orientation too, remember, because one base is going like that, the other one's like that, right? So you have to have the correct orientation, otherwise it won't stick properly. And then once that base comes in, then DNA polymerase catalyzes the formation of the phosphodiester bond. And so it uses, so if you look here, like real careful, right? There's 3 phosphate groups attached to the nucleotide. There's only 1 phosphate group in the backbone. And so this is sort of similar. It's not exactly the same, but it's sort of similar to ATP. Oh, you're gonna make me write it out, Adam. Prokaryotes. Sorry, it's real tedious to type out, like makes you feel like you have fat fingers. My typing isn't the best anyway. All right, so this looks a little bit like ATP. It's not, but it's kind of similar. And ATP has that 3 phosphate kind of group, that triphosphate group too. So in ATP, it's the last bond between the first and the second phosphate, which stores the energy. So you go from ATP to ADP and then back again. In DNA replication, and also this applies for transcription too, making RNA, the energy for forming that bond comes from breaking this second to third phosphate bond here. Right, so you break off 2 phosphates, it's called a pyrophosphate, which means you have 2. Don't know why it's not called diphosphate, make more sense. And the energy released by breaking that phosphate-phosphate bond is what's used to catalyze the formation of the phosphodiester bond. So when that happens, that pyrophosphate is released. It's just recycled, essentially. It's used to make more nucleotides or whatever, really. Now we have a new nucleotide. DNA polymerase moves on one base, same process occurs. and just goes do, 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 do. And by and large, sorry, depending on what's called the processivity, or how fast the polymerase processes along the DNA, it's in the range of a few thousand base pairs per minute. So if you have a genome of, I don't know, let's say 10,000 base pairs, and you have 2 DNA polymerases, you could do it in a few minutes, essentially. Our genome is, well, total, because we're diploid, is about 7 billion base pairs. You can now see why we have to replicate our DNA in lots of bubbles, multiple origins of replication. Otherwise, it just, I mean, you'd take like 300 years to get to being a baby, which your mother probably wouldn't appreciate. So, essentially, we have, and this is again in prokaryotes, there are 2 DNA polymerases that do most of the work in terms of DNA synthesis. Got DNA polymerase 3 or DNA pol 3. It does the bulk of DNA synthesis. Again, this is not the same for eukaryotes, right? This is one of those points of difference. And DNA polymerase 1 removes the RNA primers, which we'll get to in a second. The other ones are involved in DNA repair. Different types, presumably. Now, obviously, even when the DNA replication is finished, we're not done, right? So we have 2 kind of issues that need fixing, and there's also a lot more of this that needs doing for the lagging strand than the leading strand, right? So one issue is we have RNA, right? That's a big, big no-no, right? For no other reason that if that organism or cell was to replicate again, that RNA couldn't be used as a template for making DNA, right? It just wouldn't work. Similarly, it wouldn't be able to be used for transcription either, right? You need DNA only in your DNA, obviously, right? So got to get rid of the RNA primers because that causes all sorts of problems. And the second one is actually a pretty fundamental one. The backbone has to be sealed, right? And that's, if you think about it, right, essentially you have for every DNA molecule, you have one continuous template strand, right? Again, because it's semi-conservative DNA replication. And the daughter strand, before the whole process is finished, is a series of chunks. Now, those might be really big chunks. If we're talking about the leading strand, in which case in bacteria, it's like one large chunk, right? Eukaryotes, obviously, it's many. But we need to stitch those together. And the lagging strand is going to be tens to hundreds or thousands of chunks, right? Depending on what organism we're talking about. And so having a DNA that is fragmented is actually the signal for, at least in eukaryotes, for cells to die, right? It is a catastrophic situation to be in, right? So the cells have to stitch those bits of DNA back together, right? So that when transcription occurs, you don't, it's like basically going on along a railway and one of the rails has a break in it, right? It would be terrible, right? In many ways. Also for future DNA replication, Again, if you had a break in your DNA, you'd get to it and replication would stop, right? And it might need to start again for the next chunk, but then you'll end up with a fragmented genome, which even in bacteria would not be a good thing. So there's a whole process that has to occur to essentially fix those problems, right? And this is where the lagging strand gets its name, right? The actual DNA synthesis takes about the same amount of time. Yeah, I mean, it does take the same amount of time. It's the fixing these problems that takes much, much longer for the lagging strand because there are many, many more instances of this problem that needs fixing. Right, so that's kind of an important point to get. Most likely it will come up as a question in the exam. Almost always does in some form or other. Okay, so the first thing that needs to be done is we need to figure out where that RNA is and remove it, right? And so in prokaryotes, that's what DNA polymerase 1 does. So DNA pol 1 finds essentially what it's looking for is DNA RNA hybrid. It removes the RNA and synthesizes DNA in its place. Right, so that's generally the role of DNA polymerase 1. It's not very good at synthesizing DNA, as in it can't synthesize much. That's why it's not used for the bulk of DNA synthesis. You know, it can do like 100 or 200 bases and that just runs out of puff, basically. And the cool thing is DNA polymerase 1 does both things at the same time. So it's kind of like a pick up at the front, put down at the back, right? It's kind of picking up the RNA like at the front and essentially hydrolyzing the phosphodiester bonds between the RNA nucleotides. So it's picking those up. And it then uses the 3' end of that, the previous piece of DNA, right? So that would be the end of the previous fragment. This is the beginning of the next fragment. It uses that 3' end to add the DNA nucleotides to filling that gap. Because you don't want to have single-stranded DNA in your genome either. That's also a bad thing. And so it will proceed along that DNA RNA hybrid until it essentially removes all of the DNA and adds every single base that it needs to. So what you end up with is DNA, 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 space in the backbone, and then DNA. Right, so if we were to draw that, because sometimes it's a little bit easier to illustrate than it is to describe it. Actually, let's kind of draw it properly. Damn it, now I'm getting all pedantic with myself. There you go. Got to get the... sometimes I'm my own worst enemy. Right. So... essentially, this would be what the let's do this. Maybe kind of what the template strand looks like, right? The original one. So when the RNA primer has been removed and the DNA has been synthesized to fill the gap, oh, I'm going to have to, this is going to be going this way. See, I'm even putting the right number of hydrogen bonds in. How cool is that? And P. I'm trying to find the urge to turn my head. And then we have a gap. What would that be? T. And so this would be our 3' OH there. So this is our nick or our gap. And so though, even though every single base pair has been filled in, right? There's no single stranded DNA at all. DNA synthesis is effectively complete. In terms of adding bases, DNA replication overall is not, because without filling that gap, right, we have to make a phosphodiester bond here. That's absolutely and totally crucial. Feeling a little guilty because I just drank my daughter's coffee. She didn't have time to have it before going swimming. But she'll live. She'll be fine. She's young anyway. So this gets fixed by another enzyme called DNA ligase. Can you actually see that green? Not sure if that's clear enough. Yeah, it's a bit wishy-washy, huh?
Yeah, try a different color. I can barely make that out.
I don't have many different colors, Robert.
Oh.
I know. I'm missing. I have like all the colors in the rainbow at work in my office. And I have three. I'm like an artist with a limited palette. There you go, this is better. Black just seems so harsh though, I don't know. Anyway, so this is an enzyme that recognizes these nicks, right? These gaps in the backbone. Again, not worried about the actual bases, Cather. We're worried about this break in the backbone. And so it and ATP comes along and fixes it. So the reason why we need ATP, how many phosphates does this base have on it? No, one. This one. Thank you. Sorry, Adam. I'm being a little bit cantankerous this morning. I guess it's because it's Thursday. This only has one phosphate on it, and that phosphate is the one that's needed to make the phosphodiester bond. So there's no energy inherent in this nucleotide to be able to synthesize that bond. So if you remember, we go back up. Here we go. If you look at this nucleotide here, it has that 3-phosphate group, that triphosphate group. The energy in that bond there between the second and the third phosphate is what powers DNA polymerase. Yes, that's right. Thank you, Adam. It's our universal energy source for all organisms, as far as I'm aware. So we need to provide that energy with an external source, which is ATP, and all that it does is allow, actually, I'm just going to take that one off for simplicity's sake. Formation of that phosphodiester bond. Actually equals phosphodiester bond. When that's done, this DNA is considered by and large to be finished. Not completely done yet, because there's a few other bits and pieces that need to be finished up, mostly error checking. We'll talk about those in a second. All right, anybody got any questions? Again, now, as you probably realize, we've moved from the more kind of math and rules-based type genetics to more fact and concept-based molecular genetics. So now you're getting into the stage where you need to remember what stuff does what and why. The why helps a lot. Okay, give everybody a few moments to catch up with their note taking. Does anybody need any more time? Yep. Cool. Okay, so termination is a bit more of a fuzzy concept, to be honest, largely because it varies from organism to organism and type to type. Typically, it's when two replication forks meet. So if you think back to prokaryotes, if we have bidirectional replication for theta replication, it's going to be going around the circular genome, and at some point, it's just going to hit, they're going to hit each other essentially, right? And so then replication will be done. Sometimes there's a termination protein, but I'm not really too familiar with many cases of that. So the general rule of thumb is it occurs when the replication forks meet. Now, obviously in eukaryotes, that's going to happen a bunch because there's going to be a lot of different replication bubbles. Now, one of the key, probably most important things about DNA replication, it has to be faithful, right? And by faithful, I mean in the terms fidelity, right? And the reason for that is that whether you're a single-celled or multi-celled organism, you will undergo cell division hundreds, if not thousands of times. It's actually kind of hard to think of like an individual organism as a single cell, right? Because it divides into two, and then those are individual organisms. But anyway, whatever the case, that cell division is going to happen hundreds and thousands and tens of thousands of times. Pardon me. So every time DNA replication occurs, you want to transmit to the next generation a copy of your DNA, or if you're dioecious, you're sexual reproducing, half of your DNA. But if it works for you, essentially the idea is it's going to work for your offspring, right? Whatever form that takes. And so you don't want mutations. And this is kind of a weird thing to talk about, because typically when people think about mutations, unless they're thinking about like human disease or something, they typically think about evolution, right? The accumulation of mutations that result in us looking the way we do and not looking the way, Denisovian ancestors or Homo erectus or Neanderthals look like, right? Or chimpanzees or whatever. So, but the thing is, most mutations they either don't do anything, which is great, or they're bad for you at some level or some way, right? And so we don't want mutations to occur. We want this to be as good a copy as we can get it, right? And so there's a lot of work that goes into making sure those errors don't occur and they don't get transmitted to the next generation. Because if they do, they're then incorporated and those errors will be copied as if they're, the genuine deal, right? So as soon as a mutation gets into the next generation, it just gets copied and transmitted, right? That's how it spreads. And so the very first line defense against mutation is DNA polymerase itself. Right? And so this has what's called a proofreading mechanism, which is really neat. It basically checks its own work. I'm not entirely sure how it figures out it's wrong. I think it's a matter of kind of shape, essentially, like how the DNA fits within its enzymatic site. But if it incorporates an erroneous base, like a base that doesn't match, now this is just going to happen occasionally, not very often, probably, I don't know, 1 in 1,000 to 1 in 3,000 base pairs, right, by and large. But every so often, a base will get included that doesn't match. It's a mismatch. And that bond, that phosphodiester bond, will get made, right? So if this error isn't checked, it will get incorporated into the strand. So what DNA polymerase has to do is it has to stop, right? It can't continue. If it continues on and incorporates another base, then its ability to fix that problem is gone, right? It has to do it on a base by base process. And soon as it detects that erroneous or mismatched base, it stops, and it basically kind of rewinds itself by one base. It's actually kind of neat. It's kind of like ratchets back again. And it hydrolyzes the phosphodiester bond between the newly added base and the one that was on before it. This is called an exonuclease activity, right? So this is actually a kind of cool word. Oops, get back there. So an exonuclease basically removes nucleotides from the end of a nucleic acid polymer. And this is DNA or RNA. We're talking about DNA in this case, but we can talk about RNA later. Endonuclease. If I can spell it. That cuts a nucleic acid polymer in the middle somewhere, or essentially between the ends. And so DNA polymerase, its proofreading activity is that exonuclease activity, right? It's kind of working backwards. It can only go that away in the opposite direction to which it normally works. And so it removes that base. That base is kicked out, right? Not wanted anymore. And then it will just start up again. It will then put the correct base in, a thymine in this case, and it will just continue along, right? That increases the fidelity to, I think, about 1 in 10 to the 5, so about 1 in 10,000. Is that right? Yeah, 1 in 10 to, about 1 in 10 to the 5, give or take. Oh, I'm sorry, from 1 in 10 to the 5 to 1 in 10 to the 7. It's about 100-fold improvement. And this varies depending on what enzyme you're looking at, right? So some enzymes have higher fidelity, but slower processivity, right? So it's kind of like a trade-off between how accurate you are and how fast you are. Faster the enzyme works, the less accurate, the more error prone it is, and vice versa, right? So it's kind of a trade-off essentially. And so once that's gone, you know, the error rate is reduced, right? So the fidelity increases, but it's still not up to what you would want, right? And this obviously becomes more important the larger your genome. So for bacteria, it's important, but it's not as critical. In eukaryotes, particularly ones with huge genomes, you need to be as faithful as possible because otherwise you will accumulate a lot of changes or errors every time you undergo cell division. And so the next step, right, so they're kind of multiple steps essentially. The next step is something called mismatch repair. And so this is after DNA polymerase has gone on its way, right? It didn't catch the error that it incorporated for whatever reason, too much of a hurry. And so that error is now incorporated. Now this relies on something a little bit funky that we haven't talked about. This relies on DNA methylation. And what that means is it has methyl groups or methyl groups added to cytosines. Actually, someone asked about epigenetics a few lectures ago. DNA methylation is an epigenetic mark that you accumulate over time. So essentially, that cell, the old DNA, the template DNA, has methyl groups added to it already. It's just they're there. Newly synthesized DNA does not, right? And this is an important point to make. The daughter DNA is not methylated originally or initially, and it takes time for this to occur. So essentially there's a window, a smallish window, during which you can tell which is the template strand and which is the daughter strand. After that window, there's no way of knowing, right? So if there's an error in the DNA, you have to fix it before that window closes. Because otherwise, how do you know which is the original, correct DNA? How do you know which is the new and incorrect DNA, right? There's no way of knowing. They're just two strands of DNA. So during this window, enzymes can go, oh, that's the old stuff. This is the new stuff. This new stuff is what I want to cut out. Right. And so what happens is this mismatch is detected, and typically it's a result of a change in topography of the DNA, right? It looks odd, weird. Now, again, I don't know exactly how that oddness is detected. Don't know if there are proteins just kind of running along it like little workers on a railway track, or like, oh, there's a bump, got to fix that. Or if there's some other scanning mechanism involved. I don't actually know. It may be known, I just don't know it myself. Actually, it says it here that A's are methylated. That's it. Interesting. I'll have to check up on that. So whatever the case, whichever bases are methylated, I actually never heard of adenine methylation. But anyway, the enzymes, and these are called MUT or MUT proteins involved in mutation repair, hence the name. And so essentially these enzymes recognize a difference and they go and find the nearest methylation point, right? The nearest base in the template strand that's methylated. What they do then is they cut the daughter strand or another protein, because there's a whole bunch of them. Cuts that daughter strand and creates a nick, right, in the phosphodiester backbone. This nick then creates a free end, right? So again, remember, endonucleases cut within DNA, exonucleases cut at the end, like Pac-Man, basically, kind of going along. Soon as that end is free and it's kind of, waving around in the breeze, an exonuclease finds it and degrades a short stretch, right? Not a long stretch, because that'd be a pain, right? You'd have to fix all of it. It chews up a stretch that's long enough to include the error. And so that now is a piece of single-stranded DNA, right? That's going to be recognized by DNA polymerase. It will come in and it will synthesize, hopefully correctly this time, the bases in between, including the one which was done wrong first time round. Oops. Then that nick will be sealed and the new DNA after some time will be methylated. Right, so again, there's that window during DNA replication when the daughter strand is not methylated. That's the window in which mismatch repair can occur. If that error is not caught by that point, mismatch repair cannot occur, because there's no way to figure out which is the original strand and which is the new strand. So basically, there's a whole bunch of stuff involved making this a very accurate process. The first one is obviously base pair complementarity, right? So you shouldn't get a G matching up with a T or an A, right? Because they physically shouldn't fit properly. Like the hydrogen bonds won't work. They'll be the wrong number or they'll be the wrong orientation or whatever, right? So just the inherent intrinsic properties of DNA gives you a certain degree of fidelity, right? But it's not perfect. So proofreading enhances that, right? So it's essentially like you correcting your own spelling mistakes as you write your own essay. So that increases it by, about 100-fold or thereabouts. And then after that, you have mismatch repair. That's kind of, in a sense, getting your professor to read your first draft and give you feedback before submitting it for the final draft. And overall, that results in an error rate of between 1 in a billion and 1 in 10 billion bases. So typically speaking, every time your DNA gets copied, you make around 1 to 2 errors. Because our genome overall, if you add, both homologous sets of chromosomes together, is about 7 billion base pairs. So this, good question, Adam. So this goes on at a kind of individual per error basis, right? So the DNA polymerase, each one, whichever strand you're talking about, may incorporate some errors. Each time that error occurs, it will most likely catch its own mistake through the proofreading mechanism. And if it doesn't, if that error is incorporated, then most likely that error will be caught by the mismatch repair mechanism. So it's really a matter of probability, right? And how many bases are we talking about? Exactly, that would definitely lead to a mutation if it's not caught, right? So that kind of the point of this is saying that overall DNA replication is extraordinarily accurate through these different steps. However, it is not perfect. So depending on how many times those cells divide and how big your genome will determine how many mutations occur every round of DNA replication. But yes, if that mutation, if that change is not caught by these processes, that mutation will then be incorporated into the daughter DNA. And then the next time that DNA is copied, one template will be the original correct one. One template will now be the one that has incorporated that mutation. So that mutation will then be copied. So one cell will have the original, one cell will have the mutation, and so on and so forth. Now, the consequence of that mutation obviously is going to vary depending on where it is. And this is random too. It's not a matter of some areas being more prone to mutation than others. It's just per polymerase, per base probability, essentially. Now, there are other things that can induce mutations and DNA damage. There's actually a lot of different things that can, in various different ways. But we're not going to go too much into those. But quite often, DNA repair is not 100% accurate. So actually, as an aside, the DNA damage that you get from exposure to UV, so from sunbathing or getting sunburnt, in my case, those are called thymidine dimers. So essentially causes two T's to get cross-linked. And when that occurs, that's recognized as an error. It's kind of like a little bubble, and it gets fixed, but the fixing process isn't completely accurate, and so often introduces errors as a result. And so the mutations you get from, UV exposure aren't actually directly caused by the UV. They're caused by an inaccurate mechanism for repairing the DNA. So if we're talking about ionizing radiation, Valeria, like gamma radiation, that essentially breaks covalent bonds. And so that will induce single and double-stranded breaks. And so ionizing radiation is really bad for you because fixing double-stranded breaks is not a trivial process. And we'll talk a little bit about how that can happen in the towards the end of the next lecture period. But generally speaking, it's a bad news. And it's actually, so this is actually a kind of interesting thing to think about. The reason why, if you're exposed to high levels of gamma radiation, you lose your hair and have like lots of intestinal problems, and your skin has problems and things like that, is those are all sites of continual rapid cell division. So your hair, nails, teeth, not really your teeth, but your intestinal tract and your skin, those are all places that are constantly undergoing cell division. So if you cause a lot of double-stranded breaks in the DNA in those cells, one, you won't get cell division because those cells will die. Two, I know it's extra a little bit. If they do manage to divide, they'll almost certainly incorporate mutations. And if you don't die from that radiation, which you will if you get enough of it, you will have a large number of pretty major mutations and chromosomal rearrangements that will then lead to, almost certainly lead to cancer. So ionizing radiation is bad news. It's not like, hey, shush. Dog sitting is a pain in the bum sometimes. There you go. What a fierce little pup you are. So it's not like, you know, the Fabulous Four or anything like that, when you get hit by a solar flare and you suddenly get kind of, you know, stretchy, or you can turn invisible or stuff like that. If you got hit by a solar flare, you just die. Yeah, no Chernobyl for you. Not at all. You know, actually, just on a completely other random note, because one of my daughters did a project on this. The elephant's foot, which is basically the reactor core that has kind of melted and the uranium's like solidified into a giant lump, is so radioactive they can't actually take a picture of it because it destroys either the film or the photocell or the whatever before it's able to take a picture. Did they? But it was via using like a mirror or something, right? Yeah, they had to use a mirror. What superpower would I want to have? Oh, yeah, I think turning into an animal or something like that. Yeah. Anyway, that is totally off topic, and I need to go stretch my legs and make myself another coffee. So I'll see you back here in 50 years. I'll pay attention to you too. In 15 minutes, about 12:15, so you can have a break. My daughter Sophie wants to give you a fun fact. What's a fun fact?
Did you know that the elephant's foot is actually made of something called corium? It's an element that's only been made. Oh no!
Supposedly it's made of something called corium.
Yeah, it's an element that's only been made there.
There you go.
That's the only source of it.
That's the only place where that element exists, is in the basement of Chernobyl. That's a pretty cool fact. I think that was worth waiting for. Yeah, she knows more than me about stuff too, Adam. I wouldn't be too surprised by that. Cool. All right, everybody. See you in 15 minutes or thereabouts.